Hello and welcome to Caron Cymru and this biology session, which is going to cover enzymes, most significantly the structure and function of enzymes. Okay, so let's have a look at the essential parts about enzymes. So the first thing to note is that enzymes are protein molecules, and the fact that they are protein molecules is significant because the way in which they work is closely associated with the fact that they are proteins. So what do we mean by proteins to begin with? Well, proteins are made up of amino acids. So there are around 20 different amino acids, which are joined together in a very particular ordered way to form specific proteins. So when you put amino acids together, when you join them together, when you bond them together in a long line like this, they form what is then a protein. Now, all proteins are different. So each protein will have its own particular number of amino acids and particular sequence of those amino acids, and that's what makes that protein unique. So if you take something like insulin, which I think is about 53 amino acids long, it's not just any amino acids, it's a particular group of amino acids, and the order in which those amino acids are put together gives insulin its unique structure and function. Now, the way those, or the reason perhaps that those proteins have that unique structure and function is because that amino acid chain is folded in a particular manner. So the number of amino acids and the sequence of amino acids causes that chain of amino acids or that protein to be folded in a very particular way, which forms a specific three-dimensional shape. And that specific three-dimensional shape is different for every protein. So I mentioned the insulin protein. All insulin proteins will look the same as each other, but no other protein will look like insulin. So there we are. We go from a group of amino acids joined together to make a chain of amino acids, which then fold in a very particular way to get our final protein. Now, it's that shape which is crucial to the function. Change the shape and you will change the function. So given that the number and order of amino acids will determine the shape of that protein, and given that the shape is what makes it crucial, or is what's crucial to the function of the protein, it therefore follows that the number and order of amino acids is crucial to the function of the protein. Proteins have lots of functions. I've mentioned insulin as a hormone. They can be structural, like found in muscle and elsewhere in the body. But the one we're going to focus on is the one of enzymes, the role in enzymes. And we will come back to that structure and function of the enzymes shortly and go back to talking about the shape of that protein, of those enzyme molecules. So this is a little activity here just to keep you on your toes, for want of a better term. So you'd be welcome to stop the video and see if you can figure out what should go in each of these blank spaces, but I'll go through it right now. So proteins are made up of a chain of smaller molecules called, and those things are amino acids. Protein will have hundreds or even thousands of amino acids, and it is the sequence or the order of the amino acids which gives the protein its identity and its properties. The amino acid chain is always folded in a particular way to form a 3D, three-dimensional shape, and that's different for every protein. And that shape of the protein molecule is particularly important because shape is essential to the function of the protein. And finally, proteins have several important functions, enzymes being the one that we're going to focus on, but there are many others. So let's look at enzymes then. Let's look at their structure and their function. So first of all, what are they? So enzymes are catalysts. And you might know from chemistry that catalyst is something which speeds up a reaction, but because enzymes speed up biological reactions inside cells, inside the body, we refer to them as biological catalysts. It's perfectly acceptable to say they are catalysts, but we can do better than that and say that enzymes are biological catalysts. Let's have a look at some issues with names that we need to be familiar with here. So enzymes, they catalyze reactions and they will act upon particular molecules, and the molecule or molecules that an enzyme acts upon is called its substrate. It's called its substrate. The way it catalyzes the reaction is it binds to the substrate first, and that then will end up forming or causing the reaction to take place. But the place on the enzyme where the substrate attaches is called the active site. So you can see in this first diagram here, you can see the substrate and you can see the active site, and you can see how those things match. And we say those shapes match. We say the enzymes are specific because of the shape of the substrate is a perfect match for the shape of the enzyme. And if you change the enzyme and had a different shape, it would no longer fit. And so that different shaped enzyme would not work. So we say its enzymes are specific to the substrate upon which they work because the shapes need to match. And it's that, therefore, I say it's the specific shape of an enzyme that enables it to function. We go back, we talked about proteins and the shape, but now we're talking about this in applying this to enzymes where it is the shape of the enzyme that enables it to function. So enzymes are proteins and it's the shape that gives it its function. And so that links back to what we just said about proteins, that it's the shape of the protein that determines its function. And the shape is held together by bonds. So that whilst all amino acids are joined together by bonds within that chain, then the way that chain folds is determined by which amino acids are present, and those amino acids on different parts of the chain are held together by bonds, and that's what gives it its folded shape. Right, a little bit more here. So we talked about this is showing an enzyme slightly differently here, a different shape of an enzyme. The same principle, see the substrate fits into the enzyme. And now it's showing here that the enzyme has now joined to the substrate in this middle diagram here. And that structure which is formed where the enzyme has bound to the substrate is referred to as the enzyme substrate complex. That's a question that's asked quite a lot. What do we call that structure that's formed when enzyme and substrate are bound to each other? It's called the enzyme substrate complex. And you'll see this diagram at the bottom there. But once the enzyme substrate complex has been formed, the next thing to happen is then the reaction actually happens and the substrate is converted into products, and those products can then leave and the enzyme is free to do its job again. So it says that at this point, the substrate will be converted into a product and the enzyme is free to catalyze a reaction with another substrate molecule. So enzymes can be reused over and over again. They're very good at their job. And this principle, the way that this idea that the enzyme's active site and the substrate fit together perfectly is referred to as the lock and key theory. This is a theory that has been put forward that's widely accepted, that an enzyme and its substrate fit together like a lock and a key. They're a perfect match. They're specific. Now we say it's a theory because the enzymes and the substrates are so small we can't see this happening, but it has been proposed that this is the mechanism by which the enzyme and the substrate bind. Okay, so here's those things again. Again, you can pause if you wish, but I'm going to go through them now. So enzymes speed up the rate of a biological reaction. They are biological. The word missing there is catalysts. The substance that an enzyme works on is called its substrate, and the place on the enzyme where the substrate attaches is called, number 3, the active site. Number 4, we say enzymes are specific. They only work on one type of substrate because of this idea that the substrate shape must match the active site. Therefore, it's the specific shape of an enzyme that enables it to function with that shape held together by bonds. And knowing what's coming up in your studies, we will talk about bonds again and what the consequences are of breaking those bonds. I've emphasized that point here because we're going to need to use that in the future. When the substrate binds to the enzyme at the active site, the combined structure is called an enzyme substrate complex. And at this point, the substrate will be converted into a product, is number 7, and the enzyme is free to catalyze a reaction with another substrate molecule. Again, this idea that enzymes can be reused. And this explanation of how enzymes work by attaching to a matching substrate is called the lock and key theory. That's number 8, because they fit together like a lock and key. Everything on there are questions or facts upon which you could be questioned, and you expect to see them in past paper questions. Here's a just taking things up a little level, perhaps. So we'll do this one thing one bit at a time. I'm going to give you a definition. What are they describing? The part of an enzyme into which the substrate fits, that is the active site. The substance upon which an enzyme works, that is the substrate. A combined structure formed before an enzyme converts the substrate into a product, a combined structure, that's the enzyme substrate complex. The theory that explains how enzymes work by attaching to a matching substrate. That is the lock and key theory. Whenever you see a question which refers to a theory, chances are the answer is going to be a lock and key theory. It's not uncommon to see pupils write down the term enzyme substrate complex when the answer is lock and key theory, and vice versa. So the way you pick out which one you need is you're looking for something like the word theory. A molecule which speeds up the rate of a biological reaction, well, that is what an enzyme is, and refers to the fact that each enzyme will only catalyze one reaction. We say enzymes are specific. And a long chain of amino acids which are folded into a particular 3D shape, that is a protein, of which enzymes are one type, one category of proteins. So let's put this into what you can expect in an exam. So look how it says here, diagrams A to C illustrate the lock and key theory of enzyme action. It shows how a protease is able to catalyze the separation of 2 amino acids. So now this is not talking about the structure of enzymes being made up of long chains of amino acids. This is talking of a protease enzyme, and the fact that you see the word protease, it ends in ase, and that's your clue to know that protease is an enzyme. So this particular enzyme, what it does is it takes 2 amino acids and it separates them into individual amino acids. And I know that because I'm looking at the diagram and I'm seeing 2 joined amino acids in diagram A, and I'm seeing those 2 amino acids being separate in diagram C. So that's what it does. Diagram D shows the protease after it's been denatured, and we haven't talked about denatured yet, but I'm sure you'll see that in a future video. So, question simply asked here, what is the name given to the structure represented by diagram B? So I mentioned earlier how it's not uncommon to see students write down lock and key theory. And lock and key is wrong here, and there's a couple of markers or tell you that this isn't lock and key. The first thing in the sentence, in the starting point of the question, it tells you about the lock and key theory of enzyme action. Now they will not give you that information and then ask you to come up with that term in a later question. Well, that's very unlikely. It's certain, and they say it there, the lock and key theory of enzyme action is written there. And my guess is when this question was devised, they've deliberately put this term lock and key on twice. They're trying to dissuade you from writing it in this answer here. The only thing that was wanted here, it was that B is the enzyme substrate complex. Here's another question here that says the diagram shows an enzyme which builds up complex molecules from simple molecules. So in this case, it's showing the enzyme, which is clearly identified there, and it's showing 2 substrate molecules. So the good thing about this is I like the fact that very commonly when we refer to enzymes, we show them taking a single substrate and breaking it down into 2 products, which is what the previous question was showing. But enzymes also take molecules and put them together. So this is taking 2 simple molecules who are labeled, and they're putting them together into a large molecule. So it says complete the diagram below to show the next stage in the reaction between this enzyme and the 2 simple molecules shown above. And it's told you in the start that this enzyme builds up complex molecules from simple molecules, and it's labeled simple molecules. So your job here that's what's on the mark scheme. You need to show both substrate molecules having entered the enzyme, so I've drawn them on there, and some idea of a bond being formed between them. So that's what's expected there. And now it says what is the name given to this model of enzyme action. So I said you're looking for a word like theory.
or something similar. So this is a similar idea. What is the name given to this model of enzyme action? Now that is the lock and key model or lock and key theory. So that gives you some idea of the type of questions you can expect. That's it on this video. Hope to catch you on another one. This I hope this has been useful, and you can find more videos on the Caroline Cymru site.